At Aerie, we're developing new therapies that target the root causes of vision loss in glaucoma and retinal disease. Our lead compound for glaucoma is Rhopressa, which lowers IOP by inhibiting Rho-kinase and norepinephrine transporter. We expect to file an NDA for Rhopressa by the end of this quarter. We have another product, Rocklatan, which is a combination product of Rhopressa and latanoprost that's currently in phase 3 development. Today, I want to focus on our efforts in retinal disease, and specifically our lead compound for the treatment of age-related macular degeneration and diabetic retinopathy, AR-13154. AR-13154 is a Rho-kinase inhibitor that also inhibits the kinases PDGF receptor and Janus kinase. The combination of these activities allows AR-13154 to address multiple different disease processes that underlie AMD and diabetic retinopathy. And because of this, we think it has great potential as both a standalone therapy as well as an adjunctive therapy to current anti-VEGF treatments. Rho-kinase is actually an interesting new target for the treatment of AMD and diabetic retinopathy. In preclinical models, it has been shown to address all three of the main drivers of disease: angiogenesis, inflammation, and fibrosis. And this obviously contrasts with the anti-VEGF therapies that are focused primarily on treating angiogenesis and vascular leakage. And perhaps because of that, over time, there appears to be a significant proportion of patients who progress on anti-VEGF therapy. So it's possible that a highly selective inhibitor of Rho-kinase alone would be sufficient to address these main drivers of disease. However, in practice, multi-kinase inhibitors tend to be more effective, and particularly if one selects the kinases to have complementary activity to one another. So we went to our library of 3,000 different Rho-kinase inhibitors, and we selected 182 compounds representing different structural classes and activities, and we screened them against all available human kinases. And our goal was to identify molecules that inhibited not only Rho-kinase, but inhibited other kinases known to be important for angiogenesis, inflammation, or fibrosis. And this screening is what identified AR-13154. It inhibits Rho-kinase, protein kinase C, PDGF receptor, and Janus kinase. Now, PDGF receptor is obviously of interest because anti-PDGF molecules have been shown not only preclinically to inhibit angiogenesis and vascular leakage. The Fovista product in the clinic now has shown promise in phase 2 clinical studies as well. Protein kinase C is also an interesting target for reducing vascular leakage, and that's supported by preclinical models of diabetes. Janus kinase is an interesting target for reducing inflammation based on the ability to reduce leukocyte adhesion, and PDGF inhibition also appears to have some antifibrotic activity. So we've tested AR-13154 as a monotherapy in a preclinical model of wet AMD, which is the laser-induced CNV model. And we found that as a standalone therapy, it was very effective at reducing lesion size in this model. And in this study, actually was numerically more effective than the positive control Eylea. We then wanted to test whether AR-13154, with its multiple different targets, could be effective in combination with anti-VEGF therapy. And so we combined AR-13154 with Eylea in a model of, in an OIR model of retinal angiogenesis. And we found that that combination was actually substantially more effective than either of the two monotherapies alone. So with these experiments, we've demonstrated that AR-13154 is effective both as a standalone therapy and can be used adjunctively to anti-VEGF. Now that we've established efficacy, we're focusing on delivery of AR-13154 to the back of the eye. Our objective is to deliver it as a bioerodible implant that will need to be injected once every 3 to 6 months. We expect to establish preclinical proof of concept for long-term efficacy within the next 6 months and to begin our IND-enabling tox and CMC studies in the first quarter of 2017. So in summary, AR-13154 is a first-in-class multi-kinase inhibitor that addresses some of the limitations of current AMD and diabetic retinopathy therapies. By targeting multiple disease processes, it has the potential to provide better longer-term outcomes for patients. Through its unique mechanisms of actions, it can be used either as an adjunct to current anti-VEGF therapy or we expect will provide efficacy as a standalone product. And with our planned dosing regimen of intravitreal injection every 3 to 6 months, we believe AR-13154 could reduce risk as well as treatment burden for both patients and physicians. Thank you.